Well, welcome everyone to this overview of biologicals and what they mean, what they're useful for, and why they're a key part of the Power to Grow program. First off, I think it's important in this day and age when everybody's looking for a new opportunity to sell something new to realize that biologicals are a part of a farmer's entire crop production system. We realize that from the very beginning, and we've always promoted biologicals as a part of our crop production system. In the past, we haven't emphasized them much because prior to $5 corn, they were snake oils. And ever since $8 corn, it's the coolest thing to have. So they've always been a part of our program, and they're certainly the foundational portion of our program. And we have built everything around them to make sure that they perform in the best manner possible. So we're going to take a little dive here and got several topics that we'll have in a series of videos that we'll go through with you and share just what are biologicals, how they work, all the different facets and types that they are, and most importantly, what is included as part of the Power to Grow program and what that means for you and your farmers that you work with. So we'll start at the very, very beginning. In biologicals, there's so many different types of them. It's important to define what they are. So I think the easiest way to think of it is before, during, and after. So before biologicals are those products can be referred to as prebiotics. Okay, pretty simple, before. And biotics would be in the category that we're in. The during portion would be called probiotics, or those things that would be, let's say, life portion, those kind of things. Those are during the biological process. So pre would be inputs, food sources, those kind of things. Probiotics are the bugs, microorganisms, both fungi, actinomycetes, and bacteria that are working on those products and residues and soil compounds. Then postbiotics is the after. It's the results, the compounds that are created, the various results that come out of there, whether it's a fermentation extract and those kind of things. So I think to really kind of simplify this a little bit and help to understand what's going on with biologicals is looking at it in a pre, a pro, and a post, before, during, and after. So before life, during life, after life of the microbe. So I think prebiotics, definition: substances that provide nourishment, support for beneficial microorganisms in the soil. So these are essentially food sources. So a lot of times that comes into, various organic compounds such as sugar. Sugar is an energy source. These organisms typically are air breathing, metabolizing sugars and other compounds to live. That's where they get their energy source from. So other carbohydrates such as molasses, you hear a lot about sugar, whether it's table sugar or sucrose, glucose, mannose, sorbitols, which is actually corn. High fructose corn syrup is a biological. All those kind of things. Plant residues themselves can be components. So the sugars, the cellulose that's inside of plant residues can feed microorganisms. The prebiotics, some of the functions listed out here, they serve as food sources. But other things that prebiotics can do is serve as essentially microbial homes, electron exchange sites. It's really important. One of the key concepts to keep in mind with single cell organisms is, unlike us, we've got a mouth, we've got a digestive tract, and then we've got a way to get rid of our waste. In a single cellular organism, all of those functions, eating and defecating, have to occur across the single cell wall. So that has to be an association with some sort of an electron receptor in order to be able to bring material into the body of the microbe, which is generally at a higher concentration in the body of the microbe than outside the body. So it requires energy to bring it into the cell. And at the same time, it also has to have a mechanism to expel waste out of that organism. So having close associations with um, places that the microbe can do that, typically clays in the soil, organic matter in the soil, provide those electron receptor sites to be able to allow that chemistry to happen. Some other examples: inulin, FOS. That's a mouthful to try to say. Fructooligosaccharides, and then also soluble fiber. Other prebiotics can be things that would support the growth of those. We could say is sugar is a really popular one today. We have some various sugar alcohols involved in our products because we find that they're less likely to stimulate pathogenic species such as Phytophthora, Fusarium, and Pythium when they're essentially in an alcohol form instead of in a pure sugar form. Still provide an energy source, but less likely to provide that oxidative stress associated with breaking down of sugars. So those are kind of the food sources. The second category, the probiotics, and this is where a lot of press is spent today. So we know of individual microbes that people are selling or consortia of microbes, even some of the names are pretty funny. So these probiotics are typically going to be spore form or living organism form that farmers are applying to the soil. There's various ways to do that. The key to probiotics and their effectiveness is finding something that can survive the harsh wilderness of the soil. There's millions of organisms, species in the soil, billions by count in every teaspoon. And the ones that are there are the ones that the natural system has selected that are optimum to be there. So to overcome this optimum, there has to be a quantity effect, which is really hard to get a quantity out there in ounces per acre. So you're hoping that there's it's a better adapted and more aggressive growing species. Reality is, most of them serve as a food source for the microbes that are currently there. So it's easier to change the environment of the soil, either through tillage or no tillage, through cover crops, or through pH adjustment of the soil to change the environment. Then you will change the microbial makeup of the soil. Or it's easier to change the food source or the stimulation of the microbial environment than it is to try to sprinkle or pour in the next bug or desired species. So we'll dive into that just a little bit more. Like we mentioned there in the definition, live beneficial microorganisms, when applied to soil, enhance the microbial balance. So that's the goal. A lot of times the testing is done in sand or very devoid microbial systems or lab systems, and we can show pretty bar charts. But when we get into the real world and we're applying these things on the surface at pressures above 40 psi and water that's got chlorine in it, or on windy days and with sun, sun UV killing it, or the wind evaporating the water. And if it's not immediately incorporated or gotten it or put into the soil somehow, there's lots of opportunities for those to not work effectively. So we typically we continue to research this category. We're interested in finding probiotics or live portion that works well, but they have limited shelf life. They're sensitive to temperatures. The spores have to be kept vacuum sealed. They have to be mixed up in a certain way with a certain pH water and never sprayed out at over 40 psi. So there's just lots of handling things that is very tricky for success to happen on a consistent basis. There are, various things. We can get bacteria, we can get fungi, all those kind of things come and are available to buy. There's a lot of inoculants that you'll be familiar with, and I believe we'll dive into that a little bit later on. But one that many of you have heard of before is mycorrhiza fungi, and mycorrhiza fungi are great, but realize the majority of them are actually yield limiting because they take energy to prosper. So we have to always make sure that we're exchanging energy from the plant for the benefit that we want to gain in those categories of probiotics. The other thing that's important is understanding that when we're in a high phosphorus situation, or if we're using a very available phosphorus solution on seed, and mycorrhiza won't come to life and infect the root in order to bring nutrients to the plant in a high phosphorus environment, realize then that AMF, or mycorrhiza fungi, are essentially antagonistic to early phosphorus. So we know from years of research that and results that early phosphorus is really key for fruit set and initiation and early growth, that it's hard for us to give up on that benefit that is very consistent in order to gain an inconsistent result of AMF. And AMF really has to be placed directly on the seed, infect the root early in order to provide that. Now, mycorrhiza fungi on later developing plants may be of benefit, but especially on tree plants where you have a perennial crops, those are better applications of that product. But it's sexy, it gets lots of press and those kind of things, so a lot of people know what AMF is. And we'll dive into that a little more later. Other really common probiotics are Bacillus subtilis, and Bacillus subtilis species have been known for a long time to upregulate plant growth or help with hormone production within the plant to build a bigger plant architecture. There's also a lot of Pseudomonas species that are known for making nutrients available from the soil. In particular, we're interested also in them along with Nitrobacter that make nitrogen available to the plant. And those are key functions that allow us then to use less total applied nitrogen in those scenarios. So those things are beneficial too. And we've been aware of those things for a long time. And what we see is a lot of the a lot of the probiotic or biologicals are essentially just repackaged Bacillus subtilis and Pseudomonas together with maybe some other things to help with that plant activity. We do know that there's other ways to accomplish those hormonal and PGR related type growth factors, and other ways to get nitrogen stimulation from the parent material of the soil that we're going to dive into here a little bit later on. So the third category that we've talked about a little bit is postbiotics. So this is products that are made by biological organisms in the soil or soil microbes. And so postbiotics are either result from compounds that the microbe itself makes. It could be compounds that are left once the microbe dies, or as it splits, reproduces, and dies. Those type of functions, enzymes and other proteases are left in its place. These are degraded various structures where we have maybe simple sugars. We degrade them and complex them into longer chain sugars, those kind of things. So, a lot of organic acids, enzymes, antimicrobial compounds are produced within those are exudates, typically from fungi. Most of the antibiotics that we have today are result of finding fungi in the soil that make those, from as simple as penicillin and amoxicillin to several other the most complex antibiotics that we have today were essentially isolates from organisms within the soil. So I think it's key to know that they play a very important function. So we're not necessarily interested in the microbe itself. We're interested in what does the microbe do for the soil? What does the microbe do for the plant? How does that change? What is the role of that microbe? And essentially, postbiotics is the function that they're creating. So when you look at it, it contributes to soil fertility. We have a lot of microbes, and we learn this through essentially the recent work that Dr. White's done in the rhizophagy cycle. The plant's up is multiplying certain microbes that it needs, sending them out into the soil because they provide a function of maybe reducing manganese and making it a plant available form, or making zinc available, or any nutrient available or bioavailable to the plant. That's why the plant is prospering certain microbes to fill its needs. The plant's not interested in the microbe itself. The plant's interested in the result of the microbe. So, that's one of the things that we have to think about in the postbiotic type realm. The other things that they can do is, suppress harmful pathogens. So, like I said, in the antibiotic um, world, the various plant compounds that are made actually help protect the plant for a longer period of time than what maybe the microbe or the fungi, bacteria, the fungi were alive at that time. And they enhance nutrient cycling. Yes, it just makes them bioavailable to the plant. Rarely are nutrients taken in just the mineral state. They're taken in complex with various sugars or carbon compounds that the plant uses. So a lot of times we get a little oversimplified in what we've been taught, that plant needs nitrogen or plant needs zinc. It's what form is that coming into the plant? Oftentimes those are in carbon-based forms that are complex thanks to microbes in the soil. Other things, that postbiotics, things that would fall into, this category: organic acids, acetic acid, citric acid, those kind of things that are essentially derivatives from microbial activity. Enzymes, which we're pretty key on enzymes here. Enzymes such as, there's basic ones, cellulases and proteases, and the cellulases are fun because you can see them make a change on fibrous, and they can turn a fibrous goo into just basically liquid in the speed in which they react. There's also key things that postbiotics would be plant growth regulators that are produced. So several compounds in various plants contain plant growth regulators. So they'll have cytokinins, inylacetic acid, indolebutyric acid. All those things are produced in the soil by microorganisms that the plant then uses for cell division or cell elongation, overall growth, and increased metabolism of the plant. So what we have found historically, and one final thing to always bring up is amino acids. Amino acids are made and complex by microbes, and that make it plant available because of their activity. So one of the things that our approach has been, rather than put out bugs and hope for the best and overcome this giant pool of organisms in the soil, is let's stimulate the classes of organisms that we're interested in and provide the postbiotic, or essentially the result of source, in order to make sure we're getting the result we want in the plant. So it's a more direct approach. We don't have to put out the bug and wait for the result of the bug to influence the soil and influence the plant. We make the result of the bug, the postbiotic, in a very controlled manner, and then introduce that to the plant so that it can make the change in the soil and the change in the plant that we want. The other key thing is enzymes and these organic compounds that we're placing in the soil have far more than a single reactive life. Enzymes can do thousands to trillions of chemical reactions in their lifetime. Think of them like a catalytic converter. The platinum that's inside of a catalytic converter, it's a catalyst. Enzymes are a catalyst, and they're designed to cause several chemical reactions over and over for many, for a long service life. Okay, very similar. And so we target, and I think something that's important, you may or may not know how our enzymatic products are made, but they're made in a we control the water, we control the food source, and then there's multiple species of microbes added in consortia with each other at specific times within this, for lack of a better word, a brewing process. So we make that where we introduce 2 or 3 microorganisms, and then 2 hours later there's 4 to 6, and then maybe 4 hours later is this much at a controlled temperature, controlled duration over that feed source, and a controlled um, pH, so that way as each one of those successive generations of biologicals are introduced into this microbial soup, we're getting a continued complexing of those simple exudates into longer and longer chain carbons, and also into longer and more productive enzymes that then are at a very high concentration level that allows us to use very minimal rates per acre to get a long lasting phenological change in a plant. And many of you've probably seen Rev Up that's placed on seed or in transplant water or seed treated. You'll see throughout that plant's life, not just a change at emergence, but you'll see a phenological difference throughout the life of that plant. Because what we're doing there is causing certain genetic markers to come on, uh, to to enable parts of the plant for its own defense mechanisms, become more efficient at nutrient gathering and allocation within the plant, and create a plant structure and essentially a plant biome on the leaf and surface area of the plant, the phyllosphere. We're changing that biome throughout the life of the plant in order to make it more disease resistant, more resilient, need less inputs. But all of that's coming from a specific biological consortia that we developed years ago to improve that. And really, hats are off to Dr. Alnoff and his team that 40 and 50 years ago knew these things, and still today, it blows my mind how far ahead of their time they were. And it's been truly amazing to see how we can make a lasting change utilizing these biologicals that we use, which are primarily in the postbiotic category. We'll dive into more of those as we continue on. So for now, the key thing is to remember that biologicals can be about anything. They can be as simple as sugar. It can be simple as one bacteria. It can be as simple as one fungi, or it could be as simple as a yeast extract. Okay. Or it could be all three of those categories together. And it could be high, intense sugars. It could be molasses. I've seen whey used. I've seen milk waste, used. All kinds of things can have a positive plant impact. But what we're always constantly looking for is how do we get a consistent, easy to handle, um, less likely to get screwed up by the operator, so we have a great customer experience. And what we found is that in that postbiotic category, in particular, are biostimulants, which are driven primarily by enzymes, but also by other fermentation extracts and metabolites in there. Those are designed to stimulate the microbial community, to increase the total respiration, essentially, the total biomass of the microbial community, but also designed to increase the response of the plant when they're in presence of those stimulated organisms. So that helps bring to light. There's a lot more complexity there than maybe what we've talked about before, but I think there's a lot of people that have a shiny object and talk about the newest and latest, greatest thing, and may not know all the complexities that are out there involved with the biological community. And I think it's really humbling to know in the time that I've spent in this space, I know more than ever before, just because we're always learning every day. But the amazing thing is I'm convinced that I now know that even though I know more, I know less of the total that there is to be known. So, um, biology of the soil, of the plant, and the interconnectedness is amazing. It's an incredible design, and to really appreciate it is to get to know more about it, and just so that you know how little we do know. So, sometime if somebody's talking to you and they've got everything figured out, move on because they don't. Uh, you just they've identified themselves as a person that's not fully informed. So, so we just got done talking about pre, pro, and post biotics, right? Before, during, after. So, food sources, life, afterlife. So, Kim wanted to hop in here and ask some questions, some follow up, get some clarification on a few things. So, we're just going to have a fun little discussion about this.
Well, so I appreciate having that breakdown and the definition of those. And where you talked about the prebiotics, and those are food sources, you said,
Mhm.
for the microbes in the soil. So they are more things like sugars and things. They're not live critters, correct?
Correct. And one of the things I probably should have talked about in there is we include a lot of these prebiotic sources in our products. So, in all of the micronutrient packages, for example, they contain sugar alcohols of some type, depending on what we're trying to accomplish. So we have sorbitols on our ground-based ones. We have mannitols on our foliar-based ones because of molecular size. Mannitols are smaller. And then also we do uh, humic acid, fulvic acid, which those could be considered both probably a pre and a postbiotic because they were originally made by a microbe, and laid down in sedimentary layers, and then we react them, and it becomes a prebiotic, a food source, a microbial home for those. And we're going to talk about that a little bit later on. But those things are built into our products now, and it's just something we've always done. Haven't really talked about it a lot. On the label it says advanced additive technologies, so that's part of it. Is prebiotics in there, along with surfactant packages and such to make water availability and ability to make it blend with the other materials in there. And then in our biologicals themselves, obviously we have uh, various. I'm trying to think on the prebiotic side. Obviously, we have the enzymatics on the post. But the kelp extracts and such, those are going to be some food sources in there. Um, I think one of the, at the end of the day is they're not um, live portion. And a little bit of confusion comes out there saying, I thought you had bios, and we do. But just because um, they're not live bugs, it doesn't mean that they're not biologicals.
And so that's a good segue into the probiotics, which is that what you classify as the bug in the jug then?
Bug or spores. Something that spore just means it's dormant, ready to come to life. So a lot of biologicals will go into a spore stage when they're stressed or run out of water, run out of food, and then that makes them more hardy and resilient. Typically, those are used, for resale, so that they're easier, more likely to survive shipment and storage and those kind of things. And typically they're kept vacuum sealed, temperature controlled, preferably frozen to extend their half life. But then they're reawakened in the presence of moisture and in the soil. So
So I think that's an important thing that you talked about was that the actual handling of these products and knowing that by the time you apply them on your field that they're actually alive and able to work. And so there is some handling stuff there, and that's a certainly a
and,
concern, I would think.
and the UV thing and the temperature thing we've talked to people
Mhm.
about before, and that's why we include in throttle, draft, and turbo. Those products have a lignosulfonate in them, which is essentially functions as sunscreen for postbiotics. since we're using the new terminology now. But Revit and Restart are sunlight sensitive because that's not built into there. So, we keep them boxed or covered in totes and those kind of things. But I think a lot of that we've just known and we've always done it that way. A lot of other companies are going to learn this and learn it the hard way.
Gotcha. Gotcha. I just think that's a good distinction. So then there are no what we would call probiotics as bugs in a jug in our products. Would that be fair to say?
There is not unless
Oh, okay.
Unless you had a tote of one of our biologicals last year, or maybe even a tote of Accelerate, and you used part of it and you left it, as you drew in air, there's always microbes in the air everywhere. We're breathing them all the time. We're interacting with microbes all the time. And you inoculate your tote with a microbe, and then you will have a probiotic because you'll have something growing inside of it. It'll be floating, it'll be gelatinous, it'll be uh, not intentional, but you have an excellent food source and an excellent stimulation source. Therefore, you can introduce probiotics, and sometimes you'll notice if you draw some out or if we don't have a perfectly clean toad or just it only takes one organism and we can get a secondary fermentation.
Mhm.
So, but no,
Right.
intentionally, no probiotics.
Okay, good. I think that's good to know so that we can kind of classify that.
Can I tell you a funny story?
I would love to
So
hear
we had
funny
a
story.
customer that sent some of our products into biome makers, bee crop, really to analyze what the quality and composition of our microbes was in our product. And it came back, there was no microbes in our product. And the customer calls and talks to me, and I said, "Yep, you're right. There are no microbes in our product." And he was, he didn't know. So that's one of the reasons why we need to have this discussion and explain to people what that is.
All right. So we talked about mycorrhizal fungi, and you talked about how they won't bring pee to the plant if it's present or abundant in the soil. Am I understanding that correctly?
They won't come to life.
They won't come to
They'll
life.
stay in a spore form.
Okay.
So the plant won't allow the infection or won't give energy to that spore because it doesn't need the phosphorus.
It doesn't need it because
Right.
it's already present in the soil. The plant recognizes that phosphorus over what the spore, the AMF
Right.
would
And,
provide.
it's not going to give energy to something they don't need.
Okay, because it would require energy.
It's a root infection.
Right.
I mean, mycorrhiza means fungus root in Latin.
Okay.
And it's a fungal infection in the root. And so the plant is sending carbohydrates to that fungal species. Now, it does all the great stuff, and you've all seen the pictures of looks like a tree underground and how wonderful it is. But everything will take it. And the funny part is, I remember having a sidebar conversation with Wendy Tahiri, and Wendy is kind of all things arbuscular mycorrhizal fungi, AMF. She's all AMF. And she said most 80% plus of the products on the market create yield drag because of that energy partitioning. So we have to keep. The other thing, and gosh, there's a whole bunch of stuff I forgot to talk about, Kim.
That's why I'm here.
I know. Thank you. And that's why she gives me an outline and say, follow this. Just they put the monkey in front of the camera and see if he does what he's supposed to, but he didn't. So one key thing to know is AMF is everywhere. Everywhere. Antarctica to the North Pole, Gobi Desert to the rainforests of Brazil. It is everywhere. Now, it may not be in high quantities in desert sands, right? So you get in desert sands, you get the biggest response out of it. But in the humid Midwest or Mediterranean climates, it's there. Prosper it. And if the plant chooses to create that beneficial infection, it will. But when we give a high concentration of it and essentially kind of trick it into getting infected, we can cause yield drag. Not everything is, do this, get 5 bushel, do this, get a quarter ton. So it's important to know that there's a real balancing act in all these things.
Well, and would it be fair to say that the reason that the plant wouldn't want to get infected by it or make that connection is because everything is an expenditure of the plant. In other words, it would have to expend energy to make that connection with the AMF.
Mhm.
And so the plant is deciding, where do I want my energy to go?
Right.
And so that's the reason that it doesn't make those connections.
Mhm.
Or if it does, then know that the plant used energy for that connection rather than putting it where else would it go with it, with that energy.
Yield, root development,
Exactly.
plant architecture, all those kind of things. So
Right.
a key concept, and I don't know if it's a concept, a principle, a law, whatever you want to call it,
Mhm.
is energy. And when you apply ignition on seed, it took energy to make the ignition, right? We all know that. It took energy to transport it, took energy for you to apply it. That's one form of energy. But that's a very high energy status nutrient right there on the seed that that plant don't have to use carbohydrates to stimulate microbes or fungi in this case to make phosphorus available to it. So what we're saying is, should I buy a little bit of energy in the form of ignition, or should I buy energy in the form of plant carbohydrates? Which is the best economic return? So now, as phosphorus becomes less and less available globally, and prices will continue to escalate over the next 80 years until we potentially run out of it. It's a very finite resource. Um, there will come a time where the trade-off is such that we're willing to give up more yield to acquire phosphorus from mycorrhiza or from biology in the soil versus buying phosphorus to apply. So it's a matter of today we can buy that energy to support a higher plant yield, but in the future we may not be able to buy that energy.
Mhm.
So
Mhm.
it's a matter of where is it coming from? And that's why it's so important to look at everything in an entire system
Mhm.
of how we balance those things. And caveat, non-biological, but accelerates designed to double the effective uptake of phosphorus. RevUp's design and is shown with an enzymatic in place with phosphorus, you can 5x the uptake of phosphorus. So that work was done in UC Davis. So it's interesting to see um, how those things interact.
Mhm.
So the energy has to come from somewhere. Does it come from? Do we bring it in from off farm, or do we have the plant produce the energy?
Mhm. Let's say that phosphorus becomes less available in the market,
Mhm.
so to speak. Wouldn't it be beneficial then to build your soils, your soil health, and your entire functioning system so that you could make phosphorus available from your soil so that you wouldn't have to seek out those other avenues? Is that a fair thing to say?
Yep, that's a simple answer, Kim.
Okay. All right.
There's
But
no it depends to that one. That is just straightforward. The better your soil function is, the better the soil quality is, the better air water management that you have within your soil, the less stuff you have to buy. So if you like to buy stuff, till, put on nutrients, broadcast, um, spray the hell out of everything, great. You can buy lots of stuff. If you don't want to buy lots of stuff, pay attention to your soil, pay attention to your water cycle, your infiltration rates, your bulk density, soil aggregation, crop rotation, cover crops, all those things. Do all those be a great farmer. Do those things, and guess what? You don't have to buy as much. That's signing the back of the check instead of the front of the check, as Gabe would say.
I'm learning that that's a lot more fun. So, okay, but I think that's really key
You
is
haven't signed the back of any checks yet.
not
You're
a
just getting
single one
the
yet.
wheat
All I've
project's
signed
just
is the
starting
front.
right now. So she's signing the fronts. Anyway,
Yeah, very true. So more to come on that. But so I do think though that it is important for us to recognize that functioning soils do what they were designed to do. And so that's our role, is to get those
because
soils
look
functioning.
at the biomass we grew right here. This
Yes.
this was 8 to 10 foot tall prairie grasses that you literally could not walk through, or
Right.
that horses could not go through. And all of that was done without a John Deere plow and without nutrient ag solutions and without pattern tiling and without GMOs and without insecticide and fungicide.
Right.
How did we do that? How did we have 8% organic matter soils? And then the better question is, how do we burn it all the way down to 2.5%? That's another story. But I mean,
Well,
the
the
system
reverse.
is resilient and is designed to function when we allow it to.
Hmm. Hmm. When you talked about, you did mention my little wheat project, and we did get untreated seed, and
It's the biggest, almost the biggest wheat project in Mercer County.
it probably is.
Yeah, yeah.
So I sure know it's caused a lot of commotion. So, but we did treat it. We did treat the seed with Rev Up, and by doing that, what did we activate biologically on that wheat? What?
I don't know. Who knows? We just tried it. We tried it. No, what that's doing is again, it's Rev Up can be used as a seed treatment or as an on seed
Mhm.
in solution.
We used it as a seed treatment because it
Yep,
was the only option that we had
right.
to work.
And that's very common in small grains.
Hmm.
So it will just upregulate. It helps with early vigor and growth expression, more plant to plant evenness, more lateral branching of the roots. You'll see typically a better, thicker micro or rhizal sheath
Hmm.
around the roots. That's all the dirt that sticks to the surface of the roots. So that's an indication. You know, your goal in life is not dirt sticking to roots, but it's a visual representation of microbial activity happening at that surface. So
Hmm.
that's what it's for. And just again, a small amount, which I think was about 10 ounces per 100 pounds,
Hmm.
that can make a huge, a continuous reaction of an enzyme as a catalyst, and it
Hmm.
just constantly makes that chemical reaction to make nutrients available.
Well, and I think you made an excellent point for guys when they're out in the field and digging to dig
Hmm.
those plants and note the roots and what is sticking. Because let's talk a little bit about why are things sticking to, why is soil sticking to the roots? Because that's an important concept that fits in with the biologicals, or the biology, I should say.
Hmm. I mean, it's the exudates of the plant, but also essentially exudates of the biology. So the postbiotic process, it'll be various amino acid, glucoamino based polymers that cause small soil particles to glue together or stick together,
Hmm.
which is also the same basis of creating stable aggregates in the soil
Hmm.
to create oxygen in the soil, water infiltration, and all that free nutrition that we were talking about earlier.
And so, why do we want that stickiness and things happening on the root?
I don't know. It's more fun to drive a tractor, isn't it?
Oh, okay. All
Yeah, I
right.
don't think you want that. No,
Okay.
it's essentially nutrient availability, reducing density of the soil to where more oxygen and water can penetrate and function in the soil.
Mhm. That's awesome. So you talked about that the plants want to prosper certain microbes, and that's to solely benefit the plant, right? The plant is trying to make this connection with the microbes, so all for a very selfish reason, and that
Yeah.
is to help itself.
Well, let's say we're not the federal government and we have a limited checkbook. Okay. So you got a limited amount in your checkbook. You want to spend it as wisely as possible to make the biggest impact you can. And that plant all the time throughout the day and night is trying to make decisions on where do I spend the money in my checkbook. Well, its money just happens to be the essentially carbon that it's accumulated during the day. Where do I want to spend that? And if I have plenty of zinc and I'm making all kinds of auxins and I have no problems there, but I'm noticing that my photosynthesis is going down a little bit because my manganese levels are a little low. I'm going to prosper manganese reducing organisms to help bring my manganese back up. So those kind of signaling mechanisms are happening. And then, as farmers, we got to think about this every time we spray a herbicide or something else on there. We just whack and screw up all the signaling in the plant, and it takes some time to recover and get back to figuring out what it needs again and reallocating its checkbook. So
Mhm.
it's kind of like anytime we do something to a field, whether we till it or we spray it, it's kind of like sending the tornado through, and then plant has to find its checkbook again to start
Mhm.
writing checks.
Mhm.
So it's those events are very disruptive. But it's all a matter of the plant seeking to reproduce and reproduce as much as it can because that's how God designed it. If it didn't reproduce, then somebody else would come in and fill that void. So that's the whole purpose.
Right, right. And if you want to learn more about what pesticides and insecticides are doing to our microbial population, tune into our Jonathan Lundgren episode, which will give you the lowdown on all of that good stuff. So lots of content available for you to learn about those things. So we talked about enzymatics and that amino acids I want to make sure I understood it correctly. Did you say amino acids are made by the microbes or they are made available by the microbes? What?
Yes, both.
Both.
But
Okay.
amino acids are made by the plant. They're also can be made there. And one other thing is because that's a high energy thing and it's a precursor to a lot of plant growth factors, we include amino acids in our materials so that again, this maybe we digress a little bit, but some manufacturers took the role of going to PGRs applied to the plant. And when you're trying to size a grape or something like that, jib acid is great. But there's a popular product, Bioforge, and it has indoleacetic, it has L-tryptophan, and it has gibberellic GA3 in it, and a little bit of IBA. And sometimes you get a great response out of 8 ounces per acre. Sometimes it's a quart. Sometimes it's 16. You just never really know. It depends on the plant and what's going on at that time. So we thought that was a little risky and we didn't take the PGR approach. But we provide the amino acids that are the highest energy to produce of those precursors for the PGRs. So the plant, if it wants to, can easily assimilate and make, or for example, there's cytokinins in draft. And that's then easy for that plant to use those cytokinins. It can choose to use them or not. And the amino acids that are in Rev Up and Restart, it can choose to use them and make indoleacetic acid out of it or not. So it's those kind of things are the approach that we take is give the building blocks, let the plant decide. Plant's there 24/7. It knows a lot more about what's going on in the soil environment and in the above ground environment than I do. So just enable it to do what it needs to do.
And could you give us just like one or two examples of how the plant uses the amino acids to, is it in reproduction? Is it in, what is it in?
Um, so a lot of the amino acids are designed for stress reduction. So stress reduction, cell division, and those kind of things is what it's designed for because once a plant hits a stress cycle, it's hard for it to get out of that cycle because it requires essentially sending nutrients from the top of the plant to the root where the anti-stress components are made in the root area. So the microbes that make the various compounds that reduce plant stress are at the root tip. Well, during stress events, that's the last place that carbohydrates go to. So it just kind of, when we have a stress event, hail, wind, reproduction, heat, all those things, that plant communication from top to bottom just gets interrupted. So that's why draft along with fast track and those kind of things helps to reestablish that communication, gives it that the amino acids and the tryptophan and the prebiotics. So, the kelp extracts and stuff that are in there gives that plant the ability then to, oh, I've got the resources I need to restart that signaling back to my root. So we don't want a plant that has to live on draft every day. Now, while that would be great for sales, okay, that's not natural. So that's a high energy input. So we try to minimize that energy input, let the plant perform the best that it can within its own context. and just give it those jump starts that it needs at key timings with a herbicide or with reproduction or with a hail event, or at germination, or when we're trying to stimulate vegetative growth or we're trying to stimulate reproductive growth. Those kind of switches or events is what we're trying to give it the building blocks it needs to grow through that and overcome it.
I appreciate that, especially with those stress events, because it's not if they're going to happen, but when. And so you're giving it the tools, the plant, the tools it needs to sort of jumpstart itself again into production. And you kind of alluded to this, but that we get a long-lasting phenological effect from. I said that
Hard to
word
say,
by
let alone spell.
myself.
Let alone spell it. I mean,
That's right. I think I
yeah.
spelled it right. Didn't say it so great, but
You sure did.
um, but I think that it's important to know then that we're bringing out the best in that plant then, or activating the best function within that plant with these enzymatics. Would that be fair to say?
Yes, and I think a way to think of it is, everybody says a lot of the DNA remains dormant. Well, nature don't waste. God didn't design stuff just to be there and not used. And I think what happens is we see more of the gene code expressed in when it has the opportunity to happen. Now, we don't know why that happens, but we do know, and we'll see it in our trials all the time where we have with and without. You'll see, wider leaves, leaves that are more turgid, and stalks that are bigger diameter. It's higher in sugar content. Tassels are different colored and different size in corn. And you're like, the only difference here is something that happened 100 days ago. And so there's certainly, it's not just a pop-up or a starter. It's a season-long effect.
So essentially what we're doing is creating a solid foundation for the plant to work off of, so that when it, not if, but when, events come, the plant can be better prepared for it.
We're going to take a break and we're going to get on to the next recording.